to welcome you all to our kickoff for the fall single cell spatial analysis program seminars. We got, keep your eyes posted. We have a lot of great seminars coming up over the next few months, outside speakers and inside speakers. So it'll be pretty exciting. And then look for opportunities. We'll probably more towards the spring be having pilot project opportunities of sorts so that people will be able to compete for some funds. And so you want to keep your eyes open up for that. So keep looking at the newsletters and bulletins that we send out. And that's about it. So Nazi here will go ahead and introduce our speaker.
All right. Good afternoon, everyone. My name is Nazi Kimani. My honor and then really my pleasure to introduce Dr. Qing and me. So he came from University California, Irvine. He is distinguished professor. And then he's really CV is pretty long. It's 100 pages. You have to read it at least a day. It's very amazing person how he can do it that many research short period of time. Basically, is very briefly his education is started on Wuhan University at China in mathematics. He received his BS and Ph.D. or MS from that university. He went to Ohio State University and he got his PhD in mathematics, and also he spent sometimes for postdoc over that university. He went to University of Minnesota, and then he got another postdoc over there. And then he spent about 2 years in University of Chicago as a faculty position, and he went to California, as all of us, we want to go to California. So he went to University California, Irvine, UCI, and he spent all the time from 1999 till now. And then he was the assistant professor, go to associate, and then full professor. And during that time, he did a lot of good stuff. He was director of the several different departments, Department of Mathematics and then Developmental Biology. And then he was a chancellor and chancellor professor over there. And right now he is a distinguished professor in the UCI, University of California, Irvine. And he did a lot of work with research, basically most in single cell sequencing or analysis. Basically, he published more than 230 articles. If I have to publish one paper per year, it took me forever. So he published that many papers in very high journal in Nature Immunology, Nature Medicine, Nature Genetic, Nature Protocol, Nature Methods, Cell Reports, Cell Research, Cancer Research, etc. All is very high impact journal. He also his time spent with the student with the Ph.D. or a postdoc. Most of his postdoc is right now in the academic level as assistant or associate or professor. And basically, I wish one of his students. Basically, he got a lot of grants, NIH, NSF, etc. And then he mentored several different K awards, etc. And he got a lot of high funded grants from NIH, NSF, etc. And he was ranked And number 4, in the transcriptome, and then technologies in the whole world in past 5 years. So which is really high impact is in his work. Basically, he invented a lot of things we are using for single cell analysis, like cell chat, cell communication, and a lot of work which is I don't understand really. So he will talk about today for all those things. I will not take his time more. And then please come to podium.
Thank you so much for the long introduction. And this is the very longest introduction to my talk ever. So glad to be here and back to Middle-West. And so let me just quickly go into the talk. So my perspective of looking at biology. What did I do? This is very sensitive. I usually, I like to walk around to give a talk, but we don't have the clip. Okay, that's fine. So I look at when I'm looking at the human, in my head, it's a multiscale systems, right? And has 3 major scales: the tissue and which is organizing and by many cells, and within the cells, a lot of genetic information and the molecules. But at the center, it is cell. It's a unique aspect of life. So, and if we zoom in a little bit, and the multiscale system is very spatial temporal, right? And there's the cell fate is very dynamic, and the cell always communicate. And so they communicate in many different ways. The cell has many lineages, and complex systems of life, usually involving cell, constantly change their might not as fate, but state, and then and to be coherent, to have the function desirable functions. So only recently, the single cell omics data provide a rich information and embedded, right? It's not obvious in spatial temporal and multicellular organizations of cells, right? So for this audience, I might not need to go into the details of what is the single cell technology, but I want to say, because we talk about spatial, this is spatial analysis biology programs. So there's a technology emerge to measure spatial transforms, like very fast during the last few years. Okay, so you might have now spatial is more like 2015, 2016 of RNA-seq data. So how do you looking at the spatial data? And basically, using a nutshell is you have cells as a function of genes. Let's use a kind of abstract language functions, and with space, and they have coordinates of X, Y, Z. So basically, you have the beautiful structures, right, of the cells as a function of genes. And it depends on the technology. Nowadays, you can go to millions of cells of the genes. Is always going to be roughly 20-some thousand. So the major current efforts mostly are involving with annotation and clustering. Okay, how many cells? Well, 37 trillion cells in the human body. Is that a lot of cells? Big number? Yeah, it's not that big. If you think about it, if you're looking at the computer, right, the disk space, and so one thing I always mention is Did you know? So this number depends on who you look at. Some of you might familiar with these numbers, how big, how small it is. It's 10 to 12. So what's the US GDP? You know what's US GDP, the annual gross product?
28 trillion.
Yes, exactly, 28 trillion. Basically, it's $1, 1 cent. Okay, all right. So now you remember how many cells there, roughly speaking. So there's a lot of effort, right? Qing and work very hard to classify, sub-classify different type of cells for different organs. One major effort, believe it or not, is on brain. A lot of effort on brain to classify the cell state and the cell types. Okay, so what's the data? At the end of the day, your data is getting something like this. It's not thousands, it might be 10,000, it might be millions. That's your data. Okay? But the data is static, right? It's just like the right side, it's static, right? However, for many of you, when you look into the right side, you can imagine or even use computer to simulate, right? Before I become a full professor, all my research is on those stuff. I did biology much later. So then the question is, how we make the static thing to be more live? Okay, so this is not easy, right? So let's just think about cell plate transitions in 2 genes, okay? And for this audience, pretty much everyone knows what EMT is, right? Mesenchymal, epicyclic mesenchymal transitions. So if I work with, if you work with biologists, yeah, they have a few favorite genes which marks the epithelium. We have some genes mark the mesenchymal, and how to make transition. Suppose the whole world, whole body has only 2 genes. Then here's a potential all the possibilities. Okay, you either turn it off, the E gene, and then turn on the mesenchymal genes in this way, or you gradually decrease one, gradually add another one, or and then something in between. Either 4 or 5, depends on how you classify. So that's one path, right? So and then it's only like last 10 to 15 years people realize it's also actually stimulated by modeling. People realize there's a hybrid state, there's an intermediate state during EMT transitions. Okay, there's a lot of experimental work and the modeling work. Okay, and now if you have another your favorite quantities to measure, then you can see the path is much more complicated. Then that particular factor X could be anything. It could be morphology that we talk about today. If you look at cancer cells, it could be a protein, not a gene expressions. It could be an epigenetic marker, right? So basically, is when you're looking at a high dimension, the transition is really complicated, and so you need certain ways to quantify it, to start it. So. And usually, this is the one way people have done is, okay, we're gonna do clustering, we're gonna do lineages, we're gonna have pseudo time, and we're gonna looking at the transitions. So one of our original work is say, okay, we want this process can be described in a more quantitative mathematical language, which is, think about, it's more like a particle, which is cell, and attractors, which is a stable equilibrium state, and the transitions, which is non-stable, usually relate to the saddles, and then we want something more interpretable, and you want something more white box. Okay, so what does that mean? So basically, I'm giving you a single cell data. The goal is, can we fit in the data into some kind of a landscape, some kind of a metaphor, is you have attractors. If this is deep, stable. And here it's stable if you're perfectly on the top of the hill. but but if you perturb it, it's gonna be here. So basically, you give me all the single cell data in the high dimensional space, which is the 25,000 genes for human and mouse, roughly speaking. And then I'm going to construct something for you to describe this dynamics system. This is what we did. Roughly speaking, and we do multiscale dynamical kind of feeding the data. And I will show you a little bit of the math. And then at the end of the day, you can have landscape. And this is the neural request cells, okay? From early neural request cell, and it goes differentiate in different thickness cell. Now, this is really a 3D. It's really like here, like the mountains. Each dot is a cell. This is, of course, this is the cartoon arrow, but we can quantify it. I will show you. But before I'm going to show you, this is the only 2 slides. I'm going to show you some mathematics. Okay. So I'm not going to get into it. Basically, we're using a very old traditional method of diffusion. It's not that old, maybe 20, 30 years old. And then you think about each cell is a random walk of particles, and then you can write down the equations. You can make a transition from one state to the other, and based on the gene expressions, which is the P, and then you get the P, the probability of transitions, and then you fit the data. You fit the data, so at different scales and at attractor scales, at the individual cell scales, and then also at something in between, which are called cell attractor scales. Whereas attractor means it's a big way of holding the state. Okay, in the ideal stage, H is 1 dot. Means every cell is identical. That's impossible. Okay, so after that, and let me see. This is moving. Okay. Okay. Then you say, okay, how are you going to validate it? So one way is to generate synthetic model, right? You construct a gene regular network. For example, this is one of my previous work. This 4 or 5 years ago is the ENT system. You have the E genes, which is the blue, and the mesenchymal genes, and then this is all established arrows, and then this you write stochastic B. Basically, I simulate 20-some genes, and with many cells, starting with a few cells, then you generate a heat, which is the cells and the function of genes, just like the things I did, and with the dynamic map. Right. So now basically we have a in silico lineage tracer techniques to follow all the cells exactly in terms of its different state and its transition power. Use that, you can guide the method, right? And not guide the method, you can validate the method. Okay, so after you validate the method, so I'm just showing you one example of this is some kind of blood cells, okay, with complicated lineage, I don't know. And then we can, so for today's talk, I'm not going to dive into any specific biology. I'm going to touch many different biological systems, but I want to really emphasize the tools, okay, and how the tools helps you to understand the biology. Is those are the transition probability from the 1 state to 2 state. Those are the contour maps, basically, you just cut the landscape. The landscape has 3D, you just cut 3D. Then you can quantify all the transitions, right? So basically, if you are a system, if you have a set of single cell IC data, and you know the system has a lot of states with a lot of transition problems, yeah, that you can try this method to make it more. So I always believe when we develop this method, and I hope somebody else to use it, not because I use it. But then Wilcox method happens to be at the peak of the pandemic. And a group of people from Duke cell biology department, they work on the human lungs. And then they use our method. So this is based on our method. It's not based. We just use our package. Okay? And then they found a transitional cell. And that highly transition cell is critical in terms of which these two cell types, which is kind of a differentiated cell types to go. And more recently, of this work, and they put the COVID-19 virus on this cell type. It's very sensitive to that. It dictates how many cells goes that way and how many cells go that way, and how that affects the whole immune systems. So that's another biological story. So basically, it's I show you an example means to give you one snapshot. I can show you how to reconstruct the dynamics, right? So then we use that same idea to try to improve the RNA velocities. So for many of you might know the RNA velocity splicing and splicing informations, but one thing we know is that if you use RNA velocity, some of you have used, we find sometimes work, sometimes don't work. Even the same data set, it works on one cell types around one cell type, but it works on all cells. The reason is the RNA velocity is just one linear model for all the data. Also, they are not worrying about the transitions and the time scale between transitions and messenger RNA splicing dynamics. So we use the previous method and to incorporate basic ideas. For each attractor, we build up a different model to model RNA velocities. Our method is intrinsically has multiple time scale, and we can take care. And with that, you can also, in addition to put the data on the UMAP, our method can also working on spatial data. So basically, it's now we have something called the transition tensor, which has more information and then RNA velocity to look at transitions, both for spatial data or non-spatial data. So the example I'm showing you, this arrow, are spatial data. Okay. Take the pathways, how that affects. So these two examples, I showed the two examples in terms of method, is focused on one kind of data set, which is a snapshot, which is one snapshot. The rationale is the following. It's the system is algorithmic, means taking the one snapshot, that data can contain all the temporal information. For example, we all know the multiple, there's many different cell types in the blood. They turn over 7 days, human, 7 days, 10 days in the blood.
30
One.
days.
Okay,
2 months.
some days, okay, it's a time scale, okay. So, but at any moment, if I grab enough blood cell, all the cell types will be there, even though they do turnovers, okay, then I can reconstruct. However, for cancer initiation or developmental biology, early time and the later time, the later time might have totally different information than early time. So that means one snapshot cannot give you sufficient information. You have to do multiples, right? So now, if with multiples, so our dream is we're gonna connect them. It's almost like AI. You do a few snapshots and then I connect them. I build a functions. And previously, it's a pioneer work is done by Eric Langer's group in 2019. They did this. But it is a very simple optimal transport method by only connecting 2 temporal points once at a time. It's different. Okay. And also this is the balanced optimal transport, means the population cannot change from one temporal to another temporal. So we trying to improve that, and we build up something called unbalanced. Okay. So the idea is we're going to link all of this. And also we're going to consider the growth cell populations. So we write this in a mathematical framework. You don't be scared by it. Basically, we have a growth term, we have a transition term. And then each term is an unknown function, unknown. We have to reconstruct. What we know is the distribution, is the cell populations and distributions. So now, of course, how are you going to get these functions? We're going to write in terms of a neural network, blah, 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 ODE, which is very technical tools called dynamic optimal transfer approach. So with that, and you can do a lot of things downstream, because now you think about it, it's like I have an in silico data at all time. You give me 3 time points. I tell you at all time, including the future. Of course, how good? It depends on the number of data you give to me. Is your number of data really capture the essence? Then I can build everything in between or beyond, right? So the testing case is looking at the EMT data. Some of the EMT data has temporal information. Then the question becomes, can you reconstruct that trajectory? Yes. Right. Each dot it's a cell. Now, if you do your traditional RNA velocity, you can see the velocity go all over the place. This RNA velocity will not be able to capture this. Okay. Now, with the temporal information, so once you reconstruct the temporal information, you can do a lot with it. Because temporal information can a lot. One thing we're doing now is with temporal, we can do causalities. Okay, that's all. Let me leave that. Now, with of course, everything is built on in a context of the cells. So now it's once with the cells, then you have to, okay, I want to look at downstream, at the end of the day, you want to perturb genes, you want to look at your markers, and then you say, okay, I will do this. Now we build up a model. This is our model. We model the gene interactions with a dynamic system, with splicing, unsplicing, informations. But what uniquely we build it away, it's the GRN is attractor, means cluster, cell type. This is really can think about it. Dominant cell type specific. Okay. And then you can also figure out, because once you have cell types, you can also, by looking at the property of the model, to look at the transition genes, the genes which is critically important. Okay. So here I want to mention one thing, right? We're all familiar with marker genes, genes which are most representative for a particular cluster, a cell type or cell state. Then usually we're so wrapped up in those genes. But remember, marker gene might not be most important for transitions. We are finding a lot of cases, some genes are more important in translation, even though they're not a marker gene. Did everybody get the point? Okay, let me give you examples, right? Clearly, the hair length is the marker for men and women. Am I right? Not 100%, right? But clearly, hair is not the most important thing to switch a man to a woman. So marker gene doesn't, it only says it's statistically uniquely important. That's unique, statistically important to represent that, but not necessarily functional that important. So we're often, so when you look at your systems, don't so wrapped up in the marker genes. Oh, let's change the marker genes. Okay. So some of you are familiar with, okay, let's do market omics data. We want to looking at regulations where, where's the computing play, where the place in the computing, the transcriptomic factors regulate things, and you want to looking at the necessary conditions, which is open or close, and then you use that information to figure out, is this gene being regulated or not, and of course you have to do other experiments to figure out which one regulates. But from data analytic perspective, it's those two data sets, even though, those two data sets is very different. It's sparse and binary because it's on and off, open or close, based on some criteria. And here is more, it is a little bit high, so then integrate these two data sets could be challenged. So we did it on 2020, which is in this area, 3 years, a long time ago. And with that kind of you pair it, and then you can looking at more specific cis regulatory information to detect functional elements, and you can do a lot with it with a regulatory network. I don't want to get into details. I can show you how we're going to construct, and but I'm just want to tell you is now. I started with talking about you have static data, and you can reconstruct transitions. You can quantify it. And then if you want to know more genes, you can construct GRN specific to cell state, cell types. Then I emphasize the importance of the transition genes. That will be different from marker genes. Often they're different. And then if you want to study more about what's regulation, and then using a taxi, and we have tools to say, okay, let's look at the regulatory element. So far, I haven't mentioned anything about space. Right, so it's only time, but time often comes with a space. Okay, now then the cells all communicate, right? They communicate in space, and they communicate through ligand receptors, and often, for example, one type of cells release some molecules that can diffuse, sometimes transported by other mechanism to another location and captured by receptors, and have the ligand receptor complex and turn on the downstream genes to do the job. Okay, so so this is actually our first work, right? When I first looking at single cell data, my original goal is to figure out communication. The reason is at that time I work a lot on morphogenesis from modeling perspective, not from the single cell analytic data. Then I say, oh, you know, if you do modeling, you usually have to work with the biology. Who's the morphogen? You look at literature, when you find, yeah, it's BMP, it's wind. Is this all the morphogens? So basically, so my dream at that was, okay, can I have an unbiased way to figure out all the morphogens, right? So we first get the idea, so we're very lucky. Okay, let's do it. Let's just say we figure out who talks to whom, individual, all individual cells. So our original goal is to look at all the cells, every individual cell, how they talk to each other. So we come up with a biophysical binding model to look at how cell I talk to cell J, and including some downstream information if you have it. And basically, I give it a ligand name, for example, wind. A wind study example. There's so many different names. FGF, something which is more specific, FGF5, and the downstream receptor, FGF5 receptors. And then you just look at the expression. Then we found that there's a lot of false positive. All right. And it's, of course, about 60% are right. When I say right, you still cannot validate in the context of individual cell because nobody knows. So still in the context of cell types or subtypes based on literatures. So after that work, so we started this work in 2019. It takes a long time to publish this paper because nobody believes we can do this. By the way, the second author is here. Right. So I have a former PhD student here to postdoc. So if any questions on this, ask me. And then we say, okay, let's be less ambitious. Then we say, okay, how about we build something on more like a population level? Okay, and also I'm thinking a little bit more comprehensive in terms from the regulators of the receptors. Okay, and they consider the populations and build a more kinetic model. And cell group I total cell group J is ligand, I and J. So they got build up this model. So the postdoc started to work on it. So and then he immediately got good answers. Means the code of going from here to have everybody knows the cell chat. It's very easy. Now, what takes a long time, about 6 months to 9 months is, so at the beginning, I feel, okay, you give me as a user, ligand and receptor, I just inputted it. So my collaborator said, no, he wants us to give a database such that we can systematically screen every possible, in his language, every possible ligand receptors. Of course, they're not possible, based on literature, the known ones. So then I say, if you like to do it, you do it. I don't have patience to go through database, to literature to figure it out. That's not my strength. I'm not bad, I wouldn't say I'm not bad. I would say I'm not good at this. So then he worked for 6-9 months with my postdoc to come up with its manually plus existing database come with this mouse and human database of ligand receptors and the regulators. So he worked very hard. So I think that actually pays off a lot, in my simple idea of just giving a model and you give them, because this allows people to screen much easily. So that's why we get a lot of attentions. And so I don't want to get into deep, everybody knows something. But what I really want to say is, finally, this paper is out for people who wanted to know more about how to use it. It's a Nature Particle paper coming out a few days ago. And it takes a long time. Then one reason it takes a long time, I can tell you for this audience. At the beginning, I'm very ambitious. I said, what's the point of just do a tutorial almost? Particle is just a tutorial. How about I build a new method into called cell chat 2, cell chat spatial. We build in one of the reviews just said, no. I can only allow you to publish whatever published before, but you just give a lot of details how to use it. So then we go back and forth to fight with it, and then we have to pull out all the spatial analysis spectrum. Okay. So basically, for some of you might already use it. So I know once we put on the GitHub, people just say, you can use it. And now it don't allow us to publish. So yeah, okay, anyway, we're working on that. So that means that's one caveat. So, but the key here is, the study of the new here is looking at systematically to give ways to do comparison between two different conditions. That's not in original cell chain. Means you have disease control. One way to do you do them independently, you look at the eye, what's the different one? Oh, now we have systematic way, a lot of tools here built in, okay, to compare with disease and non-disease. So let me tell you, just give you a couple of examples how we can use it, because today, then we talk about wound healing. So one thing we did is use this as an example, but wound healing is it's an easy experiment. I designed it, yeah, it's old and young. People knows the old mouse doesn't heal as good as the young mouse. After you give a cut, watch the functional outcome. Then during that healing process, we just sequencing it. And then after we sequencing it, we do comparisons. Right? So my idea, very naive at the beginning, was that I don't run that. So this had to convince my collaborators to do this. And I said, I guarantee that output, either the same or different. Either way is interesting. I said, yeah, they did it. So then I give you 50K to do all this experiment. So now, why the 50K is important, I will come back. Then what we found is, for example, this mouse, roughly speaking, for somebody might know, it's more than 60-year-old man. Okay? It's older than me. Then you're looking at the young and the old. So there's so many different cell types because we just unbiased rough. So there's immune, there's fibroblasts, epithelium, all kinds of dermal and epidermal, it's all there. Now if you list all kinds of cells, okay, but trust me, the cell is about the same, young and old. Even the proportion, if you look at it closely, they're not far away from each other. Means old and young has the same cell population with a similar size. But did you see the difference between this and between this map and this two? Did you see the difference? This is all aged and young. Did you see the differences? The cell population, yeah, the same. For people who have run cell check, you immediately realize there's much more links here. So, cell talks a lot for old folks. But the outcome is not good. The output is not as good as young. It's almost like, oh, let's do something more bad, and I'll do it. Let's do it. I still cannot do it. It's almost like that. Okay. So why people keep asking me why? I can give you a lot of intuitive systems biology, mathematician hypothesis. But once we start to do experiment, we realize, oops, we need to kill a lot of the old mouse, which is very expensive. So that's the 50K, just stop there. So that's why we can only publish in cell reports. So we cannot do functional analysis or more perturbations. That's the key. Can we do perturbations? People keep asking. We in the paper, if you read it, so this paper is starting to really get people's attention. It's cited a lot now. And we have a lot of good small indications, basically predictions or hypotheses, whatever you want to call it. But we did not dig into it. So now you can pick up something to dig into it from web lab perspective, not from the computation perspective. So that's another thing. After that, we're looking at it's a physician. It's a director of diabetes center. Of course, you look at insulin-related aging, insulin resistant, sensitive. Then what do you see? So let's do the most crude cell chat calculation. It's the number of communication links. You don't worry about what signals, okay? So you can see what the old insulin resistant has the most communication. The old and the young, both are insulin sensitive, still old. Somehow the old guy just want to talk. Okay, that's observation. It's unpublished, this work, right? We're still trying to figure out a lot of things here. Some of them are related to metabolisms. And this is the work published in joint with people here. So I have a lot of friends in this, but somehow they're all not around today. So this one is not around today. So we have a joint effort between the dermatologists and a group of people at UC-Irvine on skins. It's a collaborative, organized effort. So it's not like old and young now. It's your hips, your palm, and so 3 parts of the body parts. The skin, if you look at the not strengthening, clearly palm for whatever reason. That's the most communications. For example, for nudge. That's another way to use at different parts of the body, look at the differences and similarities on the communication. Okay. All right. So the final one example I'm going to show you, which is more intuitively trivial to understand. If you're looking at carcinoma and the control, you're looking at the TNF, which is immune-related signaling. This basically is control. This is the tumor. Of course, tumor has more communications. Of course, it's going to be it's going to impact with the immune cell. This is because this is immune-related network. So those after doing that, then the next question is, can we move this to space, right? So basically about 1 year ago, 2 years ago, we said, okay, one thing we did not consider, even the model of that paper, is kind of a competition between receptors, different receptors and different molecules. Once you occupy, you cannot occupy again. So can we consider that? Another thing we did not consider, we did not have spatial information there. Can we do once I have spatial data? Can we? Yes. So what do? We use something called collective optimal transport. Basically, think about there's a group of cell has ligand the group cell and the receptor, how they match it. Okay, so and then this is simulation data to show you it actually works well, and then we tested on few technology, right? BSM, slide stick. What is this? It's a birdfish. It's a birdfish or. So I'm just trying to say we cover like 3 or 4 different kind of spatial transform technology. I think this is birdfish. So then this arrow are generated by our method. There's small arrows there. Okay. They're looking at the cell communications. Now, the nice thing about this method is it is, of course, based on resolution of your data. It can go as fine as single cell if your data is single cell. Okay, so the other question is this is really open up a big kind of area about how to use how to design different optimal transport method to take into account different constraints and the properties for data integration. So optimal transport, let's give you something extra. It is a great tool to integrate spatial data. Okay. And so we come up with something called supervised OT. So then last month, after all this, okay, then I'm coming to this more interesting biological questions. At the end of the day, is like, I talk to you using one language, for example, Chinese, and then you might talk to him using Spanish. For some reasons, means communicate. In sales, I can talk to you with BMP, and then you decide to say, oh, I'm going to like my neighbor to do something more highly proliferated work, which is usually wind. Oh. So what we have done is we construct the causal information to almost like activate inhibitors. We figure out a signal, inflow, outflow, they're different. Remember, cell chat, we can only say the following. Say, I figure out I talk to you is wind. And the cell chat cannot tell you is after I tell you what you do. Here basically says, I have an inflow, then I also with the inflow, might cause some outflow through this gene, those group of genes. It's like, okay, those group of genes. And the different, this is a whole mouse at different locations, has different gene modules. For example, this is the BMP, different FGF. We go through those gene modules, talk to SHH to reset. So this is very useful for signal relay. It's very useful to uncover signals in space for organizations. Remember, cell chat is very simple. Only talk about what talks to what. Okay. This now finally has a causal. I don't want to use the word causal because it's too strong. So that's why we use the word flows. It's intercellular flows. Okay. So after this, then you're working with the neuroscientists and no. And the neuron talk. In addition to traditional ligand receptor like wind, big molecules, they also with a lot of small molecules, neurotransmitters. Okay, that neurotransmitters might have at least 300 or 400 different kinds, and that is gonna turn on certain. That gonna be facilitate sometimes together neurotransmitter. Okay, so. Then we come out with something called neural chat, which is very neuron specific communications, both for spatial data and not. So all you need is onic data or spatial, some kind of spatial data, no fish, this or sick fish. Okay. So let's keep going. That's finally, we're facing this problem. And if you do a sequencing of a tumor, the non-tumor cell, you are not sequencing. I talk to the tumor. That means the tumor cell might do something, might have some gene expressions, which responding to the signals, external signals, which is not measured in your data set. So, for example, if you grab the epidermal, you sequence it, right? But the dermal will have some signal, in particular, those cells near to the epidermal. Can you deduce? Yes. The answer is yes. You say how? Just by going through the downstream response, right? Means if the cell responds to wind, however, there's no cell within the measured cell produce wind, then that's the signal either come from cell or come from outside of the room, which is not measured. Right? So basically that's the principle we use to systematically use the for external signals. So cell-cell communication is in terms of computational tools, it's a fast-growing area. So this is a tree reviewed about 9 or 10 months ago. So I reviewed the papers. So they're beautiful. So I basically I said they give a beautiful table. I said, yeah, can you do something better than a table? When they come up with a tree, that's beautiful. But I think this is not my idea, but I force them to think. Can you? So they come with a beautiful tree to think about which method in which branch. I think it's a beautiful way to summarize results. And but my point is not about tree. My point is about this tree might be twice as bigger now, even within the last 9 months, a year, right? So it's a fast evolving area. How many minutes I have? 5, 10, 20, 2 hours? Or 5?
About 3 minutes to finish and some time.
Okay. Let me finish. Let me just 3 minutes to finish. Okay. So let's come back. So now the goal is, for instance, all together, right? So one system we started wound healing, and we found for some cases, this is a mouse, the large wound, actually is better without a scar or without the scar. And you say, wow, how is that possible? So we collected a lot of data, and from single cell to spatial transform to functional imaging mutant. So my collaborators really aggressive. Take that data. So he says, go Qing, go analyze it. Oh my gosh. So basically this is challenges. I want to use this as examples. I think many of you are going to face it. One's getting more data. One is spatial temporal integration. I already talked a little bit. Another is multimodal data, ataxy, spatial, non-spatial, metabolic. And then batch fact. This machine, that machine, student A did 12 months ago, student B, which is much more brilliant, and did again, or decide to do more. And 3D informations. And we talk about a lot of mechanisms, hypotheses. Can we think about a better way to link the data to hypothesis, right? So basically, we're in a time, for many of us, is we have to put everything everywhere all at once. Okay, so one thing we have done is how can you remove a batch effect without removing biological intrinsic variabilities? So we have some effort. I just want to show you this method. At least you should remember the name, because I think we are the best in terms of remove as much of the batch effect as possible. So we did a lot of standard analysis compared to some of the standard method. For people who have done batch removal, you must have used the SRAT. SRAT is a highly over aligned. Means in order to make sure we can put us in the same UMAP, then removed a lot of difference between you and me. which some of the difference are really difference between you and me. Okay, so very careful. So okay, and we integrate the data on the wound healing. It's not our data. Many different people's data. In particular, look at the fibroblasts. So we had a conversation yesterday. Fibroblasts. Oh, crazily dynamic fibroblasts. But there's many different data sets. We integrate them, then we can deduct a lot. Okay. And the final thing I want to emphasize is, I think this is going to be here for a long time. You have a beautiful imaging data with 1 gene, 2 gene, and some of you might have 40 genes or 40 different proteins, or some of your epigenetic marker, spatial temporal, beautifully. And then you have this beautiful RNA-seq data. Oops, this is one snapshot. Fixed animals. So can you connect them? Yes. So we have solved this problem actually a long time ago in a relative sense, or 5 years ago. So we published this paper, which is massive called Spot OPSC. It's highly used now. People start to really realize the importance of this because there's more of those data now. There's more of this data now, but none of them is fully satisfactory in terms of all the information you want to get. So once that, you can do a lot of things. You can do, for example, this is old. You can look at 84 imaging plus RNA-seq data. You can get the early angle of this Drosophila, which is look at where communicating what use DPP, and DPP, which is like BMP, and that windlass is more like wind. So you look at the communications. And the final is, you want to be unbiased. You say, a ligand receptor pair is great, that's applied knowledge. But sometimes there might be the communication molecules. I keep coming to me, the biology. Oh, your database might not be all sufficient. Okay, if that's not sufficient, then we can do unbiased. So I don't want to get into the technical details. It's a deep learning method. And then we call something space flow. We build in something called, which is very appropriate for this audience, is we call pseudo spatial temporal map. Right? Everybody knows the pseudo time. Now I put spatial information there, specifically for the purpose of analyzing spatial data. And you can do 3D now, no time. So let me finish up by saying, the dream is always going to be, you can generate right hypothesis by doing exploration first, and then to testing to find something useful for disease treatment. So this is one example. Is we actually using cell chat for communications, putting a model. This is simulation of computer. And then one interesting thing for this particular work is we found something about senescence cell actually can do some good things. In senescence, is always considered to be bad things related to aging. And we found something to be useful, but also related to cell communication. In particular, senescence cell can secrete some information to stimulate regeneration. Sometimes, not always. So I believe we are in a new era of biology. If you really want to fully understand the system, you want to start something more unbiased, which is single cell RNA-seq data, spatial transcriptomics, and then to generate, to find something more interesting with testing. Then you can generate a model to see how those information connected. It is the one you expected, or to generate some hypothesis, and then you're testing, and then you might want to go back this circle. Okay. I feel finally we have the technology here, and we are there, but we need to do it. And so let me give a quick summary, right? Talk about the transitions, which is temporal, communications. It's about space, spatial temporal. And then I talk about, you're putting all the data together. There's a lot of other issues. But I really want you, for this audience, to pay attention to these two tools: space flow, which is pseudo spatial temporal map, This is linking non-spatial data with non-spatial data use OP. Okay. So those two tools can really help you to fully utilize your spatial information. All right. I think we are in a revolution of biology. It's single cell biology, it's spatial biology, and one of the challenges is also tremendous because of high dimensional data, right? But I know in particular challenge for people in medical school, you can generate data, you can tie it up with beautifully with the disease questions, and you can analyze this. But here's my slogan: you take the data, we'll make the data great. Okay. Again. Okay. So let me thank all the people who did the work. Sorry, Matt, I didn't put your name because I gave you a special. It's here? Oh, it's here. Oh, I feel nervous. I don't want to miss any of my students, postdoc who contributed to the part of the talks I did. So sorry for being late and thank you so much. Yeah, thank you. Yes.
So that the knowledge base from those endocrine factor or paracrine factor can be the basis of the modeling.
Yes.
But when you think about those non-coding RNA or extracellular vesicles, the metabolites, how can you coordinate those data into your modeling?
Yeah, my pipeline, I told you yesterday, but it's a secret. We're working on it. It's under review. You'll be invited back in a year or two. I'll give you a long story on it. MicroRNA was one group in China did it. And I don't know if it's published. I reviewed it for them. Okay. Long story, I know there's some people working on it. Go to read. Okay. Basically, there's many different communication modes. Right? You can communication through liquid receptor. I talk a lot. But my talk is quite different. Right? And all the communication through blood. And through many other extracellular vesicles. So people suddenly realized during the last year or two, you can use RNA segregator to address all of them to some degree. So in that review paper, they give the list of what are the communication modes. So, but here's my philosophy. So you say, how could that possible? Basically, so this is my philosophical perspective looking at RNA-seq data. Any mechanistic thing working communication must involve a group of genes. Did you agree? Must. Okay. That means something happening or not happening is strongly correlated with that group of genes. Is it? Okay. Something happening is communication happening, not happening. Must correlate. So, but you can always argue, well, it's a protein, blah, blah, blah. Of course, if gene's not, there's no way you get it. So based on those fundamental philosophy, you can predict infer anything, if any communications, if you know the communication mechanism, how that associates with what genes. Okay, that's one angle. In my opinion, it's prime knowledge-based bias angle. Okay, the basic one. Another angle, that's what we're working on too. Is don't worry about genes. I just want if cell A depends on one. But that's a tougher question. If I depend on you, means some causal information I can deduce. That means you must. That's another angle, which was not knowledge-based. Okay, I have a lot of this, of some biologists strongly believe internal gods is why the organ branch in certain way because of certain communications taking place. Those communications could be through blood, yes. Could be through other mechanisms we don't know yet. But he gave me he gave me a challenge questions. If I give you two organs and measure spatial transform data, these two organs are not far away from each other. Okay? Can you unbiased without looking for liquid receptors or this knowledge-based database to figure out are these somehow talk to each other? Of course, that's a very challenging question for me. So she's just specifically related to some part of graphs, some part of code or whatever. Data.
So try to have that some discovery from the data.
I hope, right? But it's not going to be easy because then you can always argue how direct the causal relationship. Because causal is always not easy. So that's why at the end of the paper, I put a premises on causal. So we are facing, if you talk about the biology, you say causal. Causal means a lot of magnetic details. So A interact with B, they give you C in a very specific biochemical ways. That's causal. On the other hand, if I say gene A high, gene C high, but I know it's not gene C high correlated with gene high. I know A lows, C lows. Did you call that causal? So there's a lot of different definition about causal. What do you mean by causal, right? So the most biological relevance causal in my language is perturbation causal. If you say, I perturb A, C change. But don't perturb A, C doesn't change. That's a causal, even though there's many intermediate steps I might not figure out. Do you agree as a physician?
Yes, yeah. And if you think some pathway is region, then you do the knockout of the gene, or they put it back and the rescue experiment. So then you can have the several data set showing that causality of the molecule.
Yeah, but it depends on who you're asking. It's very natural, but I was thinking perturbation is the way to test the validated causal.
Yeah.
But if you talk about statistician, that might not be. There's 3 or 4 popular ways to define causal. Okay, but here's the good news. Finally, the data started to become available to talk about the causal now. 10 years ago, it's very hard to talk about causal, in a systematic, unbiased way. Yeah. Oh, yeah, sorry.
It's really, it's like really fantastic tools. It's like really exciting to see like how much information you can extrapolate from the snapshot static data about the dynamics. My question is, like about the you use like direct net and inference and the like regulatory elements and like also like I think Tigon also involves some like regulatory networks, but like eventually my question is like if like essentially there's an internal some like ODE that
no, we did not put ODE in that model.
Oh, I mean conceptually, like
Okay.
not specific models, like you can like use some information to like regulate each other.
Yes.
And help them predict some like
Yes.
dynamics for the next
So-
one.
so-
Just like wonder whether you have actually like tested some of
No,
the.
we haven't. Yeah, we haven't. But I think some other people has tested. Not necessarily in a way, I don't know exactly. Basically, so her question is, you talk about gene regulator network 1, 2 interactive. How about we just put the dynamic system ODE if people know ODE? And then we just infer the parameters and we say, okay, we have a quantitative description of this, right? I think people have done this in some fashions, but we did not do it. But we did it for the other one, splicing on splicing. It's ODE. It's a simple splicing model. So in principle, yeah. So here's the also it's kind of wide work because you can think about each cell is a sampling point. Then you have 200 cells or 1,000 cells for one cluster. You basically have many sampling points. Then you can fit the parameters, right? So basically, if you write the equation it has 20 parameters, I have 2,000 cells. With 2,000 data points, then of course I can fit. It's just a matter of how good and how you match the fit. Once the fit, you can tell how they talk to each other. Yeah.
Very nice talk. And I have a naive question. I'm very sorry about this. But we talked about, I guess, dynamics of with snapshots of information. Right? And it was a very nice correlation with your EMP data experimentally and predicted from the snapshots. So what that means is all the snapshot, or be it even the experimental data, which by itself is a snapshot at a particular time point, endpoint measurement at a time. Are there ways by which you can measure transition between those endpoint measurements in a sense?
You mean, when you say what to measure, you mean doing experiments?
Either experimentally or predict?
No, predicting, we can.
For instance, the reason I'm asking is, if I were to measure something across the circadian rhythms, right? I
Okay.
can choose 2 time points, both of them during the day, and I would probably get the same endpoints.
Oh,
Because-
circadian would not be a good example because your snapshots. Let's you have to use our second tool, man. Your snapshot will not capture all the information.
Yes,
So you need is the 12 points from morning to morning, and then I connect with you with the beautiful periodic. So by the way, there are a group of people, at least 1 or 2 group of people, who are working on single cell circadian data to extract the periodic info, to extract the period or whatever information. Yeah. We did circadian. To look at the immune connections. Yeah.
Sorry, you need to go to
Oh, go to
the
the.
appointment to 2:15. I'm so sorry. If you have any question, you can talk. to
Okay,
him
thank
after
you.
that. Thank you so much.
Thank you.